For those of you that don't know me, I'm Patrice Fort. I'm a neuroscientist, associate professor at the University of Michigan. And I have been privileged to work with this group, part of the MTM Vision Initiative, for a couple of years now. And I've been directing the Biorepository and Resource Center that we've been working on setting up for the past year. And I'm going to give you a relatively quick update relative to this effort in the next few minutes. First I want to acknowledge the group Robert mentioned. We've been incredibly lucky to be able to hire a great group of people to manage that, and so I just want to make sure I recognize them. So Kelly Ramos was the project manager. Corey and Carmen, who are tremendous technical help for this project. So our goal, as Robert mentioned, is to really help create a world without vision loss from diabetes. And I'm not going to spend a lot of time on this, but just reiterating that this is one of the pieces of the initiative where we want to provide the community with a globally accessible resource linked to a collaborative network sharing their data in real time on a dedicated queryable data platform. And the goal, obviously, is to identify new therapeutic targets at the molecular and cellular level. For this, we've established the Biorepository in the Bram Tower of the Kellogg Eye Center. And really with the goal of better understanding DRD at a much higher level and facilitate the cooperative identification of novel therapeutic targets. So as I said in the last year, we have been able to set up the Biorepository and establish a team of incredibly talented people and trained to advance this project. We've assembled all the specialized equipment necessary and that are currently actively used. And we're working based on standard operating procedures that we've developed with our collaborators over the last 10 years. And that we've shared with our new collaborators that have been added to the program. The program has been onboarded with the UM Central Biorepository, CBR, and where we are transferring and storing all the samples. So over the last year, we've increased our recovery by adding 7 different eye banks that are covering 21 states. As I said, all of them using the same SOPs that have been developed over time. We're collecting medical information and complementing them with in-house phenotyping, including fundus and OCT. I'll get into that a little bit more. We are recovering from both type 1s and type 2. As of today, we have collected 16 samples so far. Those samples are being processed and transferred to the Central Biorepository, as I mentioned. And as proof of concepts, we've published already 6 papers over the years in top-tier journals with some of our collaborators, some of you being here today. So as a way of representation, hopefully it shows a little bit better on the screens over there than it shows on my screen here, but those are images that have been taken on those samples that we've recovered through the Biorepository, and where we are able to do fundus photograph, as I mentioned, but also OCT and OCM, OCT microscopy, where we can look at specific features in a detailed way. And the goal being that we all know how diabetes and diabetic retinopathy presents in a very heterogeneous fashion. And we are, among other things, really pushing to get more specific analysis of those features and better understanding of those features. I had some great discussions yesterday already with several of you relative to some of those aspects. And here is an example of a snapshot of a fundus photograph on the left and the corresponding OCT on the right. And if we do cross sections of those, we can actually look at the features relative to this area specifically. And the goal is to select those areas and push the analysis on a molecular level with specific understanding of those regions. Following up on this, once we have recovered and collected those specific areas, what we're working on currently is to perform more specific and molecular analysis. Here is just an example of a double immunostaining for different markers of the vasculature, but obviously this is just one representation. We're very interested in better understanding all aspects of the retina, and so we're combining specific targeted analysis like this one to the development of spatial multiomics analysis on those cross sections as well. But the primary analysis that we're doing on the control lateral eye, so everything I showed you so far is done on one eye that is fixed right at the time of recovery and processed in all those ways that I mentioned. While the control lateral eye is fresh frozen, allowing us to do more broad and multiomics analysis. And what we've been doing is to perform analysis on the different regions of the retina. We had some discussions before relative to the different presentations of DR, and which really led us to isolate separately the fovea from the macula, from the peripheral retina, as well as the vitreous, to correlate clinical phenotyping corresponding to those donors to multiomics analysis, including transcriptomics, proteomics, and lipidomics, to get a broad assessment of the changes that are taking place over the course of the disease and the progression of the disease. More recently, we started pushing this analysis further by starting performing single-cell analysis, and I'll get into that in just a minute. But relative to the bulk analysis, obviously the primary goal is to perform multiple types of analysis, cluster analysis, of the unbiased cluster analysis to look at specific changes that are happening in the different regions of the retina as a function of the progression of the disease, but also identifying overlapping and region-specific changes going to the level of the pathways all the way to the genes, to identify those unique and common pathways that are involved and associated with the progression of the disease along the whole spectrum. As I said, we recently started moving into more cell-specific analysis, doing single nuclei RNA-seq analysis on just a couple samples, and this is just a representation of this preliminary analysis that we've done on 2 of our samples from that have been recovered so far. And we were really excited to be able to identify all cell types of the retina in those analyses. And actually already, with just those 2 samples, see some validation of some findings that have been done and reported in the literature or in collaborator labs relative to changes specific to certain cell types. Obviously, that will require a lot more work. And this is just preliminary, but the goal is to really get to a large assessment of those changes in a cell-specific manner even farther than what we've done so far. So those of you may have seen this slide before, but I wanted to sort of almost finish on this. The goal of the Biorepository and Resource Center is really to provide a platform so that we can identify new therapeutic targets, as I said, through analysis of those common and region specific retinal changes. as a function of the progression of the disease. Potentially biomarkers identification, whether it's through imaging or through molecular biomarkers that would be identified through multi-tissue and multi-stage correlations. And then follow up on this with target validation and target relevance assessment. We want to make sure we're working on the right targets, and those tissues that we're collecting will help us better make that assessment. And as part of the initiative, all of this is being built, as Robert mentioned, through the elaboration of a platform, a web-based platform in collaboration with John Holmes at Penn, who's been developing this already for nPOD and for KRT1D with the goal that this will be usable in a large way and translatable between those different platforms. So such platform would present in part like this, where filters and diverse parameters could be searched for and allow all members of the initiatives to look for the type of donors, cases, groups that we have gathered over time. And that will be updated in real time as we are continuously collecting samples and allow you to look for and identify what is available and what can be requested. So I want to finish with basically next steps for the biorepository. Obviously, we need to expand this. We need to build a large sample set of highly characterized tissues, and we're working on this, including through collaboration with other people that have already worked on aspects like this. I had a great discussion with Toke yesterday, for example. George that will be part of that panel in a few minutes. We're really, the whole point is to make this a collaborative effort to really help move the field forward. We want to also establish a level of correlation between retina and blood and retina and vitreous. There were some comments relative to this, including by George. And where we need to understand how the vitreous can be or the blood can be used as a surrogate, and what is available more systemically and reflected potentially. We are obviously interested in going deeper and deeper in the analysis, going at the cell-specific level for these alterations and better understanding those. And finally, as I mentioned, build this platform for easy access and search of donors' information, as well as the data themselves that have been generated, but also identification of samples that are available for collaborative projects. With that, that's in a few minutes the update that I wanted to give you on the biorepository. And I want to, before we move to the panel section, I'm privileged to ask Mark Atkinson, who has been leading nPOD for over 15 years and now expanding with KRT1D. Mark has been an incredible supporter and incredibly helpful in setting up the biorepository, and we're very privileged to get his take on this aspect. And thank you very much.
Oh, thank you, Patrice. I want to just again congratulate he and his team for the remarkable progress they've made over the last 2 years in terms of seeing this formed. So, and again, it is an honor to have been the founder of the nPOD network, and we'll be explaining that in a few minutes. But I think the overall messages I want to give today is trying to learn lessons from the past to, again, move complications research moving forward, and the central role for biobanks in that process. And I want to start actually with some examples of how the impact of biorepositories have had on type 1 diabetes and our understanding of it. And there's many different types of biorepositories, one of them being serological. A lot of these studies began in the 1970s when we first began to understand that type 1 diabetes had autoantibodies, which are a biomarker in and of themselves. And they were in general population and in studying family members. And then the NIH formed this large study called the DPT-1, and which became TrialNet, and then a group called TEDdy, which was to look at environmental determinants of disease. And each one of these ended up being large efforts with over 500,000 individuals involved. And they allowed for so many new advances in terms of serological biomarkers that predicted who was at risk for the disease, time toward type 1 diabetes development. On the right-hand side, together with ADA, JDRF, FDA, we were able to develop our own staging model to define the state processes that go on to develop disease. Now, where are we in 2023? Actually, we're trying to do general population screening because that's where 85 to 90% of our new cases occur relative to family members. And we're now had our first FDA-approved therapy for delaying type 1 diabetes onset, again, all dependent on serological biomarkers. Then there are genetic biorepositories out there. Again, these began in the 70s with studying individuals and their family members, doing a lot of pedigree studies, plus studies of twins and triplets. And again, a major effort began, though, in the early 2000s, the Type 1 Diabetes Genetic Consortium, which involved analysis of about 35,000 people. These type of studies allowed for genome-wide mapping of loci that form susceptibility or resistance to the disease. And, the latest on this is that now whereas we once thought that type 1 diabetes genetic risk might involve a handful of loci or less, now we're up to over 80 risk loci in the human genome. But it allows for a lot of studies in terms of how genes influence disease, mechanisms of action. And the other thing is these genetic consortiums did open the door for big data biorepositories and open access. So, and there's a huge future in the genetic biorepositories. But the one that we started was this nPOD, which is a network for pancreatic organ donors with diabetes. Studying the pancreas used to be was popular in the 50s and 60s when autopsies were common, but then through the availability of animal models of disease and the decline in autopsies, the field kind of stalled. So in the but these biorepositories, and I'm going to share how they work in just a moment, they've identified for things like disease heterogeneity, the type 1 diabetes autoimmune disease. We now know targets in the insulin-producing cells that are targeted in the disease. We now know through studies of the pancreas that actually the individuals with type 1 diabetes have a smaller pancreas than age and BMI matched individuals. At the time of onset, it's 25 to 35% smaller, but then within 2 years of disease onset, it'll be a third to half the size of a normal pancreas. So these come through pancreas biorepositories. And all of these efforts complement studies of living subjects. They're not meant to replace, but we think that they add value. And we'll be moving forward with the design of next generation therapies thanks to them. So nPOD, again, founded in 2007 based on a strong history of where the studies of the pancreas have brought us a long way from the studies in the 1800s on the endocrine-producing cells, later pathologists showing, gosh, there might be two diseases here, what we now call type 1 and type 2. They went through evolution of names. It was more than just age. So thanks to funding from initially from JDRF and then from NIDDK and the Helmsley Trust, we were able to form this where we actually, and I'll explain, where we obtain tissues from organ donors throughout the United States. And actually I have to thank the University of Michigan and the BREM Coalition, a number, Bill Brem here, who's a very loyal University of Michigan individual, that the early stages of this, so along with Chris Rhodes here, we discussed how this could optimally work. And it actually does have some ties back here to Ann Arbor. And again, Dr. Kusmartsiva, who's in the audience, oversees this program today. And so how nPOD works and how we design it is we work with, there's 56 organ procurement organizations, or OPOs, in the United States, and we work with every one of them, either directly through contract or through intermediaries. About 2.5 million people die every year in the United States, but only about 14,000, just short of that, become solid organ or tissue donors. But we not only identify those with type 1 diabetes, or in particular recent onset disease, but in addition, we screen people using these biomarkers that came from the serological banks to identify people actually before they're diagnosed. And I'm happy to say that 2/3 of the organ donors in the United States under the age of 30, we screened to see if they were in that stage of type 1 diabetes development. But it's a big process, and it takes a lot of individuals. We have up to 17 people on call 24/7, 365, and a lot of moving components here. And that, Patrice's presentation, I think, gave you a bit of the flavor of the complexity that happens here. But the organ procurement organizations contact us. We evaluate the suitability and what program this individual their tissues would best go for. And then it starts wheels a process where the tissues themselves, they arrive in Gainesville, Florida, where the University of Florida is. They're processed by a team. Some go into storage, some go out for fresh tissues. They're distributed all throughout the world to approved projects. And then we have a way, like when we were hearing about data coming back and data sharing, we try and obtain data from the end users and do our own analysis. And this is a busy slide, but there are only a couple things that I want to point out. And I'm happy to say maybe we can actually add the MTM initiative, the biorepository, but we think that nPOD actually paved the way for a whole series of other biorepositories in type 1 diabetes that are based on tissues, and they're in that first area there. But in that, it also allowed for expansion. We don't just collect pancreas, we collect spleen, thymus, intestine, heart, kidney. We actually work with a lot of different tissues that are important for understanding diabetes. We, again, are supporting something on the order of 320 projects in 21 countries around the world. We have tens of thousands of stored samples, and but it takes time for these to develop, and that's one of the things I've talked to Doreen and Robert and Patrice and others about is, again, we've had more than a 15 years head start, and now, nPOD has over 600 cases in its biorepository, and then other related biorepositories put us up closer to 1,000 sample pancreatic tissues, whole tissues. But it just takes time and an evolution. And so, and again, to Patrice's talk, I'm amazed at how quickly in just a couple of years they've got it to. But, it's more than just the numbers. It's what did they do to the dogmas that I'm most proud of nPOD in our efforts, is that it's changing the way we treat our patients, and hopefully with time, it'll even change the way we speak to them. So for example, all across the United States today and around the world, if somebody's diagnosed with type 1, they're going to be told, oh, you're here because 85 to 95% of beta cells are destroyed. That's not true. You can have as 50% destroyed and develop symptoms of the disease. There's the whole, and then another thing they're told as well, within a few weeks to months, all of your beta cells are going to be gone, and you'll have essentially just an exocrine pancreas, and that also is not true. Dr. King here has colleagues, and they've looked at their medalists 50 plus years, and you can still see in some individuals insulin positive cells. And then I don't want to have the time to go through here, but the whole notion of disease heterogeneity, that type 1 diabetes is just a singular disease. These are the types of dogmas that, again, thanks to the studies of the pancreas, that we're able to overturn. Oh, and then there, one more I'll mention is that most people thought that it was just an autoimmune disease and that the beta cells were essentially innocent bystanders. Well, now we know that beta cells themselves contribute to the disease formation. But in the last few slides here, the big frontier, I think, moving forward is in the area of disease complications. I would say it's a safe bet to say that for decades, the vast majority of complications research was performed in animal models of the disease. And if you sit down on a microscope with the pathologist, we will tell you that in many instances, what you see in animal models is not reflective necessarily of the human disorder. And we think that there should be a rollout of these complications tissues from humans. And we've started one and worked with the directors and leadership of NIH, NIDDK, and NHLBI to create this new program called CARE-T1D, which is Cardiovascular Repository for type 1 diabetes. And, you know, we think that together, lessons learned from it, along with the MTM Biorepository and we're really going to push the field into believing what I say is true and make changes. So CARE-T1D is, like I said, a central and standardized cardiovascular biorepository full of SOPs, and it's formed to generate tissues not just from type 1 diabetes, but type 2 diabetes and those without diabetes for comparison by both in-house studies and end users. The end users are involved tissue distribution, so these biospecimens and all of the data from approved investigators. So individuals have to submit an application. It's peer reviewed, everything real rapid. But and then these tissues are distributed actually free of charge to individuals in order to, again, improve research in human type diabetes. And then the other thing that we're very active in, and it's very much like the working groups that Robert described, and Martin, in that what we did, we do annual basis is meet with our scientific advisory board and say, what are the key questions and unique features of this biorepository that we can address? Here's how it works, again, and I in part explained this earlier, but we work with the OPOs to identify cases. We receive somewhere on the order of 700 calls per year. So of those 13,000, I and my team receive about 700 calls. And then we make go, no-go decisions on a case. If it's a go, it comes in, we receive the tissues noted here. This is listed for the CARE-T1D program, and then Dr. Kusmartsiva and others, they're all processed. It's organized chaos, but because you work things as rapidly as you can. And we process tissues not only for the studies that we're going to do, but we perceive those on the outside are going to do. And then the rest of the process goes like most in academia in terms of knowledge gains and publications and presentations. You've heard the notion of the working with John Holmes at UPenn. This has been an amazing feature because, and this just complements Patrice's slide, but there's, again, our goal is to try and get as much information out as possible, and it includes IRB, privacy included information or recognized case summary information, case history. What medications was this individual on? Age, weight, BMI, all the clinical history that we can obtain from the medical records we put in here. We have the data, visualization tools, and then shopping cart. We actually worked on like Amazon, and you can go through, look at slides, these 60,000 samples, and pick what you want, and then everything's automated with emails. Another thing I just want to point out before we end here is the nice thing is that when we started nPOD, the predominant way of studying tissues was on the left here, very classical immunohistochemistry, hematoxin and eosin staining, but now, as we heard earlier, it's AI and it's new technologies and single cell. And so it'll be interesting over time. And like I said, at time, I think the UM effort is getting a good start because we didn't have these technologies as part of nPOD at its start, but as new things come available, we integrate them in. And it's just, and make the data available, and it's expanding it. I'm not going to go through this list, but this is just a partial list of the things that Patrice and I and his team began to talk about 2 years ago. And again, it's just so impressive to me to see how many of these issues or roadblocks that were there at the start have been effectively handled. And with that, I just want to say that from the beginning, we were warned that most biorepositories fail because they're museums. Or they become museums, meaning people have a great idea, we're going to make a biorepository. But what happens is samples go in, but either through personal agendas, bad performance, no QA, QC, a whole bunch of reasons, the samples don't leave, or the information doesn't, the data doesn't get shared. And again, I think that from what Patrice and his team are putting together, this won't be a museum-like effort, and I think it's going to be very active in terms of improving our understanding of eye disease and diabetes. So thank you very much for having me, and I look forward to the panel discussion.
Thank you so much, Mark. Please, I'm going to invite all the panelists, George, Chris, Fabio, and Judy. Okay, so musical chairs. So I wanted to start by, so we had Mark's perspective with his long-term experience relative to nPOD and now KRT1D, but with this panel, we really tried to get a representation from all avenues, and so I'd love to get to start by getting the point of view of the other panelists that we have today. So maybe I'll start with George because of his experience as well in this context and his interaction. And George, what do you, if you can tell us a little bit your perspective in the first place, and then maybe what you see as the priorities for such projects?
Thank you.
Yeah.
Yeah. So thank you for giving me this opportunity and great effort. And Mark, so nPOD has been incredibly helpful. But I just want to start off give a example how important biorepository is. I think many of you are very young, so you probably don't remember back to 1993. Okay, so this is, let me give you the story how Lloyd, Paul, Ilo, and I were able to identify VEGF being a critical, important factor for PDR. So the factor X has been talked about, I think, since 1948 for PDR. And due to the discovery of Napoleon Ferrara at Genentech of VEGF, which is 1992, Lloyd Paul was actually in my lab that time, we decided to determine whether that was important for PDR or not. The reason we were able to finish essentially in 6 months is because Lloyd at that time already have started to collect over 200 samples, vitreous samples, from all over the country of different type of eye diseases, including PDR, vein occlusions, and so forth. So on top of that, our lab's been studying retinal endothelial cultures and what makes it grow and not grow, high glucose, so forth. So we're able to use those two technology and repository essentially identify VEGF as being one of the most important factor for PDR within 6 months. So this is the real power of biorepository. So I think using this, we could move earlier, as you said, to identify what's causing early disease of DR, because different stages are very different. that leads to be my next point. Now, we've been thinking about this a lot for many years. So another important repository is really you need to get samples from multiple stages of diseases, because each stage of DR are very, very different. And I truly believe mechanisms are different. So not only is number of samples do you need a diversity of samples, type 1, type 2. So there may be very differences. I could tell you that cognitive decline for type 1 and type 2 are very different on pathology. We have collected 25 brains from people with type 1 diabetes. They're totally different from type 2 diabetes. So again, different type of diseases. And lastly, I do want to, let me finish up here. I think the analysis is going to be so important. I mean, sharing of everything is critical, as Mark has shown really, really wonderfully with nPOD. But analysis of data of these omics is so important. And for each end organ, I truly believe the analyses are very different because, again, it's not just, what's different, what's the FDR number, what's the p-value. Those are helpful, but they are actually not that helpful. But understand the disease process, what pathway are important. You're going to get 20 pathways. How are you going to follow up, right? So again, the consortium could bring an expertise here for the eye into really understanding what pathway shall we follow up, validate. So these are really, really important things. And biorepository is critical, very hard to do, very expensive, and very hard to collect. But it's so critical.
So lots of points to bounce on right there. Chris, do you mind actually bouncing on that last point relative to the analysis and prioritization, for example?
Sure. I should mention just at the outset, I have a confession to make. I am the director of the bioinformatic core at the University of Michigan. And as such, I'm seeing the world through the lens of data and what you can do with data and what you sometimes can't do with data. I think with regard to, an essential challenge in biorepositories is having the right samples. And I think that both Mark and Patrice spoke eloquently to the importance of having the right samples and having the right protocols to collect them. I think another challenge is, that the scope of what you can do with those samples in terms of the analysis is changing. It is dynamic. People's level of comfort with those different analysis approaches is changing necessarily. So in effect, what data repositories are trying to do is build a bridge between the researchers and the data, but neither one of those points is fixed. You're trying to build a bridge between two moving targets, which is extraordinarily difficult. But I also think that something that Mark alluded to, actually both Marks alluded to this, was the notion of harmonization and the idea that these data repositories are generating new types of data that are not organically compatible. They are not necessarily computable, which limits people's ability to actually translate those into actionable outcomes. So there are logistical efforts that are really important to make those types of data harmonious. And I think there's philosophical shifts. And I think, Mark, you had in your slide, moving from me to we to them. Okay, yeah. As long as we end with us, yes. I think it's good. So yeah.
Thanks, Chris. Fabio, your perspective from?
Yeah, so thank you. I actually would like to provide just the drug development perspective, like Dolly did before. I think that it is extremely, and I totally agree with what Professor King said. I think that what is really important for us, and I would just maybe to give not only the perspective, but just the example. We're currently working on a diabetic retinopathy program, and the first candidate was kind of dismantled, was not followed in 2010. So I think that we are 13 years away from the first drug that was not followed. Now we did it with a traditional method, and would have had, if we would have had a biomarker or a repository, something that could have helped in selecting the compound earlier, we would have been much ahead, maybe just for from Judy's perspective, to have the drug go to patient first. So it's definitely very important to have first locate the biomarker, the surrogate markers that are of human relevance. We are still looking into animal diseases and animal models, which are suboptimal. Otherwise, we would have not so many failures. And we definitely hope for our new compound. But the point is, we need a different way of developing drugs that will take into account the correlation between the biomarkers and the functional effect, saving the site, what actually is the effect, what makes a difference for patients. And we would need actually to know it much in advance. And this will help us in selecting the compound and the dose and all the development plan going forward. So the human biorepository will definitely did what probably Napoleon Ferrara had for anti-VEGF, and we need to repeat this this on a larger scale. So that it was I think that it will be really important to follow up on that. And the last point, from an industry perspective, we're collecting data in clinical trials. But for obvious reasons, we are not allowed to collect banking of samples, and especially genetic. I think that this can be done only from an independent, university, research-oriented institution. So this will be important because this is something that it cannot be repeated. We need to treasure it and not have it as a museum, as Professor Atkinson said, but just to have it as an ongoing source of important data for decision making.
Thanks, Fabio. I think, highlighted a lot of the different points and really motivation for this. Judy, I'd love to get your perspective from the patient and family side, and what you'd like to see, any comments you have relative to this effort.
So I am a layperson with no scientific expertise, but I'm a mom of a daughter diagnosed at 2.5, who's now 41, and has some very serious eye issues. I don't care if it's a Tesla or a Rolls Royce or an old Ford Pinto. I just want to get there. And I guess I get a little bit overwhelmed with listening to all of the discussions and going to breakfast or lunch or cocktail reception. And I listen to you and I hear, oh, gee, I think this would work. Oh, the data in, sometimes someone's careless and we make these assumptions and all of these studies go out based upon faulty data. Or, the litany of things that you all say. So I guess my hope is that each one of you become a heretic and that you forge forth on the best collaborative data using real human tissues, because animal models aren't going to get us there. But then you take a step back and you look at your gut and you say, does this make sense to me? And it doesn't matter if it makes sense to Lloyd, but it doesn't make sense to Chris. It doesn't matter any of that. You need to put your own twist on this, because once we can get an informal atlas of all of you and all of you out there of what you think might work based upon the data that we've collected and are collecting, and you read that, and it's not in a computer database or whatever. You just read that and you start thinking about it. That's where I hope we go. And sorry if I'm the heretic here, but I just want to get there no matter what. And if we start looking at every single pathway, every single protein, every single gene, every single thing, we just get in a quagmire. And let's just get there.
Thanks, Judy. Yeah, I think that sort of highlights one of the points that Robert made before, which is we're all here for that, is to challenge each other and figure out what is the best path forward, and really trying to make sure we're doing the right thing and we're putting our best foot forward to actually make a difference and really do something that makes sense. And so it's not just a single person's decision, but rather a community trying to identify how we can really move along with this path, and really sharing with each other our past experiences and our common knowledge to actually identify what those next steps have to be. And that's a big part of why we're here today in this workshop. So I'd love to get feedback maybe from the audience as well, in terms of any comments or questions you have.
Chris German, I'm a type 1 diabetic. I have a vested interest in this, but no immediate expertise. Question for all of you as you're putting together this biorepository, and Dr. Chan kind of alluded to this earlier, biases are incredibly important. And as you put together and collect tissue, how are you assessing the biases that are being built into the repository, the biases that are inherent to the data that will come out of it, and how are you communicating that to the clinicians when they're developing or assessing biomarkers and developing endpoints?
Yeah, maybe I'll get started a little bit, but I'm more than happy to get other panelists to talk about that. This is going to be it's a large effort, and as you said, biases are inherent. So we're trying to control in many ways every aspects of the process, get as much quality control as we can at every step of the way, and doing things like this involving other people to actually ask those type of questions. Okay, what can we do to complement that? So I just had a comment a little bit before by Wiley about, okay, what gender, what races are you collecting from? Are you getting really the right information relative to this? And that's the type of feedback that we're looking for to make sure that we are accounting for all those, or as many as possible, I should say, of those biases and those additional and other questions. We had Dr. Chang mentioned relative to the AI and having to really have the right training cohort so that it's applicable down the road. It's the same thing with the biorepository. We're going to need to, and we're trying to expand. That's why we're working with not just one iBank, but really collecting broadly to get as much representation as possible. We're not anywhere close to what Mark is able to do at this point, but we are working on this. So yeah, it is something to constantly be aware and constantly striving to improve as we go with back and forth. Yeah, George.
I think that's an incredibly important question, but it's really down to validation. So right. I think a diversity of source or material can get around the bias, because each repository may have its own bias, but if you go 3 or 4 or 5 repository, then you could possibly get rid of that. You know, for our VEGF story, I mean, within a month or two of our publication, essentially 5 to 10 other vitreous sample repository confirmed the VEGF elevation in PDR. So I think a diversity of a repository, and then you have to do validation. So validation of different living people with different samples and so forth. So validation is critical, and this is also with the omic and all the omics, you really have to do validation, which I have to admit, I noticed recently many of the omic study people are not, even in very top tier journal, are not doing validation. I'm not totally against animal studies. So I think for mechanism and interference, really interrogating what mechanism involved, then you have to go and do culture cell animal study, then come back to human. So essentially, we get the initial information from human, then validate it in the wet lab, and then this is the classic traditional clinical study. I don't know if people remember this. Everybody says bench to bedside. You know, the classic tradition is actually from patient to bench, and then back to bedside, right?
Thanks. Arup?
Yes. I agree with George. A very valid point, and also agree with Judy. Many times we study animal models to, study a disease, and many times we get excellent results in preclinical studies with a drug or a particular molecule, but they never, many occasions, I would say, not never, that doesn't get translated into clinical trials. The example of aldose reductase story. There's so much work on the animal models, didn't get translated into successful human trials. So, although animal models are important, but I think we should start from the human disease. From human disease, we find a molecule or a pathway, and then see if this is important, and study in animal model. My point is that we learned today from George that how the vitreous VEGF levels in 6-month study, led us to a conclusion that VEGF is important for PDR disease. I think it's also important to, so vitreous samples should be, my request would be that vitreous samples should be collected from harmonizing or from the different phenotypes. We know that we get vitreous samples only from the PDR mainly. It's hard to get from DME patients, but we should try to get as much as samples, vitreous samples from all over the country to have it, for the investigators. But also important to know that diabetic retinopathy is a heterogeneous disease. Not everybody develops PDR, not everybody develops DME. And there are many patients who are protected even in spite of long duration or HbA1c levels. So it's very important to study the genetics and epigenetics. And for that, in addition to collecting the eye samples that we are doing now, we should also collecting the blood samples, the DNA studies, so that, and those patients should be all harmonized, not just clinical diagnosis, but it should be all recorded and standardized with the imaging from a reading center, and then look at. So I would request that these initiatives should also look into the genetic studies, epigenetic studies, and collect blood samples.
Yeah, thanks, Arup. You want to comment?
Make just a clarifying comment about the animal models. I'm not saying no animal models at all. I don't think any of us are saying it. It was, it's just that I think for many, many years there was just a strong bias toward animal models because there's certain questions you just can't answer with humans. I mean, you need animal models, knock-in, knockouts, et cetera, and in toxicology studies. But I'll give the example, and it's one that always gets me in trouble, and people like Judy and Carol Oxenreider, who's in the audience, know this. For decades, we, in type 1 diabetes prevention and cure, studied a spontaneous animal model disease, the NOD mouse. And I published dozens of studies into the NOD mice, so I'm not hypocritical. But the challenge is like I said, we have one FDA-approved therapy for delaying the eventual onset of type 1 diabetes. But in the NOD mouse, we calculated and filed all the papers. There's over 700 ways you can prevent or cure diabetes in the mouse model. 700. And over 20,000 people have undergone clinical trials to prevent or delay type 1 diabetes in the last 3 decades. 20,000. And again, most of the time, they were, on most occasions, it was based in large part on studies from mouse models. And then it was like, oh, this worked, we'll try in humans, the drug is safe. And it didn't translate well. And so I think what I'm trying to say, and maybe the others, including Judy, is I think we need to push toward, again, better understanding of the disease in humans and just increasingly use that for identification, like your vitreous sample.
Yeah. Thanks, Mark. Yes?
Was that me? This is, this is Emily Hartnett, from Stanford. No, I think that diabetic retinopathy, diabetic eye disease, also we know that there is a lot of influence from other conditions, right? Cardiovascular disease, hypertension, things like this. When you do your biorepository, do you get this kind of information regarding the tissues samples?
Yeah, that's a great point. So we're trying to collect, as Mark mentioned, also for nPOD, but we're trying to collect as much medical information as we can, and clearly we have representation of hyperlipidemia, hypertension, all sorts of other potentially confounding factors, or at least information that we need to collect, and we're keeping track of. To the extent of what we have access to, obviously, but this is a very important point, and we're definitely trying to collect everything because, yeah, I completely agree with you. It's already a complex disease to start with, but when working with humans, you are dealing with all sorts of different backgrounds and other pieces that each of those owners have gone through. So, trying to control for as much of that as possible. Yeah, Chris.
Yeah, Chris Rhodes, AstraZeneca, but still an academic at heart. I'd like to, I think, biorepositories are fantastic. They're really good for understanding pathogenesis of disease, and they're really fantastic for what in my side of the fence now we call human target validation. They're absolutely brilliant, and I love all the big data sets that has been accumulated. But a slight concern I have is that we silo ourselves into different tissues, like we have the eye guys, the kidney guys, the heart guys. And I really implore that you come together, particularly microvascular problems, right? Which is sort of the opposite of retinopathy problems. If you come together, you'll learn more about the pathogenesis of the disease. Also, who's susceptible for getting a particular complication of diabetes, and how fast might they progress with that disease. These are really important questions that we're just on the cusp of addressing at the moment. So don't silo just the eye, talk to the other guys too. And I realize omic integration is really a big deal and really difficult to do, but I think we might just be going down rabbit holes if we don't talk to the other guys too.
And that's a great point, and that's something that we're grateful to be doing with Mark, and trying to really not get siloed like this and think about the problem more globally. We're running out of time. Maybe one last quick point question.
Michael Chang, National Eye Institute. Patrice, Mark, I loved your talks and learned a lot. And George, from talking at the reception last night, learned a lot from that. My question is that I love what you're doing, and it seems like what you're having as a repository of data and tissues, for people to have access to. And I think what happens in the real world is that people have these and do different analyses of, one gene, one pathway, whatever it is that they're studying. And of course, there are people who study in animal models, that one gene, that pathway. And I wonder if sometimes, people may have trouble looking at the big picture of how their one gene fits into the overall pathway of the pathogenesis. And so, of the disease, which is really what we're trying to get to. And so my question really to those of you who've thought about this, have you considered including pathways and results of studies in your repository? In other words, would it be useful to have, who has tried what? And I don't know if it would be a graphical representation of all the different things. Maybe it would include eyes and other stuff. And I just love your thoughts about whether the repository should just be limited to data or whether it should be results of analyses from data.
Absolutely. Chris?
Yeah, I think I have a hope that we're at the first wave of a different kind of maturity of data repositories, and that that some of the newer functionality is not just going to be capturing the information about the sample, but capturing the molecular or the clinical assay information that was actually part of the experimental design. I think we've already seen success in cancer, looking at the Oncomine database and the C-Bio platform and the TCGA, if folks are hip to cancer. So I think those are good early indicators in that other disease area. I think it's critical that we find ways to capture those results so that we can combine what you described as individually maybe underpowered experiments. I would maybe recharacterize that as focused experiments that could be repurposed at scale. So I'm really hopeful that we'll see that secondary wave.
I think that's incredibly important. I just take a second. The reason is diabetes is actually a metabolic disease. Last week I had to remind the nephrologist that diabetes actually is a metabolic disease. We have abnormality of insulin, glucose metabolism. The reason I mentioned this, if you draw an inch around glucose, there are 50 pathways involved. And so we absolutely need what Michael just said, a repository and analysis of different pathways that's changed with different organs, different tissues, photoreceptors, vascular cells, RPE. So all these pathways and cells are regulating their fuel metabolism differently. And so without what you're proposing, we're not going to make a whole lot of sense at the basic pathogenesis.
And if I could just add, Matthias Kretzler speaking next. He's going to tell us all about what he's doing.
Yep.
Well, he's a nephrologist, let me mention that.
Well,
Okay, thank you very much to the panel again.